To make the lecture shorter and shorter. Okay, let's study about the gram positive cell wall is very thick and has extensive cross-linking of the amino acids side chain. In contrast, the gram negative cell wall is very thin with a fairly simple cross-linking pattern. So let's start it. Figure 4. Here is figure 4. The gram-positive cell envelope has an outer cell wall composed of complex cross-linked peptidoglycan, teichoic acid, polysaccharide, and other protein. The inner surface of the cell wall touches cytoplasmic membrane. The cytoplasmic membrane contains protein. So here is like gram-positive cell envelope, peptidoglycan layer, cell wall, and cytoplasmic membrane, embedded protein, and that span the lipid bilayer. The bacterial cytoplasmic membrane, unlike that of animal, has no cholesterol or other sterols. An important polysaccharide present in the gram-positive cell wall is teichoic acid. It acts as an antigenic determinant, so it is important for serological identification of many gram-positive species. And we can see the diagram, figure number 1.55 number figure. So about this, here is written: gram-negative cell envelope has 3 layers, not including periplasmic space like gram-positive bacteria. It has number 1, cytoplasmic membrane surrounded by and a peptidoglycan layer. In addition, a gram-negative. In addition, gram-negative cell has a unique outer cell membrane. Inner cytoplasmic membrane, as in gram-positive bacteria, contains a phospholipid bilayer with embedded proteins. Gram-negative bacteria have a periplasmic space between the cytoplasmic membrane and extremely thin peptidoglycan layer. This periplasmic space is filled with a gel that contains proteins enzyme. The thin peptidoglycan layer does not contain teichoic acid, although it does have a small helical lipoprotein called murine lipoprotein. This lipoprotein is important because it originates from the peptidoglycan layer and extend outward to bound the unique third outer membrane. This last membrane is similar to other cell membrane in that it is composed of 2 layers of phospholipid bilayer with hydrophobic tails in the center. What makes it unique is that the outermost portion of the bilayer contains lipopolysaccharide LPS. So you can see here, gram-negative cell envelope, lipopolysaccharide LPS, and then porine, and then porine, and outer membrane. Here it is, outer membrane. Murine lipoprotein is here, it's like a spring, and periplasmic space here, and embedded protein is here, cytoplasmic membrane here. So here is chapter O, specific side chain, core polysaccharide, lipid A, and a toxin. So it says, for this, lipopolysaccharide LPS is composed of 3 covalently linked components. 3 covalently component 3. Okay. Outer carbohydrate chains 1 to 50 oligosaccharide units that extend into surrounding media. Thus differ from one organism to another and are antigenic determinants, this part is called O-specific side chain or the O antigen. Think of O for outer to help remember this. The center part is the water-soluble core polysaccharide. Anterior to the core polysaccharide is the third component, lipid A, which is a disaccharide with multiple fatty acid tails reaching into the membrane. Lipid A is toxic to human and is known as the gram-negative endotoxin. When bacterial cells are lyzed by our efficiently working immune system, fragments of membrane containing lipid A are released into the circulation, causing fever, diarrhea, and possibly fatal endotoxic shock, also called septic shock. Embedded in the gram-negative outer membrane are porine proteins, which allow passage of nutrients. These are also unique to gram-negative organism. We will see the chart later, but here is some what does this mean clinically, bacterial morphology. So what are the names? So what does it mean clinically? The difference between gram-positive, gram-negative organism results in varied interaction with the environment. The gram-positive thickly meshed peptidoglycan layers does not block diffusion of low molecular wet compounds, so substances that damage the cytoplasmic membrane such as antibiotic, dyes, detergents can pass through. However, the gram-negative outer lipopolysaccharide containing cell membrane blocks the passage of the substance to the peptidoglycan layer sensitive inner cytoplasmic membrane. Antibiotics, chemicals that attempt to attack peptidoglycan cell wall, such as penicillin lysozyme, are unable to pass through. Interestingly, the crystal violet stain used for gram staining is a large dye complex that is trapped in the thick cross-linked gram-positive cell wall, resulting in the gram-positive blue stain. The outer lipid-containing cell membrane of the gram-negative organism is partially dissolved by alcohol, thus washing out the crystal violet and allowing the safranin counter stain to take. Summary of difference between gram-positive, gram-negative bacteria. This is 1.7. Okay, gram-positive cells, gram-negative cells. Gram-positive cells have two layers: inner cytoplasmic membrane, outer thick peptidoglycan layer, 60 to 100% peptidoglycan, and low protein, low lipid content, no endotoxin, low lipid content, no endotoxin, no periplasmic space, no porine channel, vulnerable to lysozyme and penicillin attack. Gram-negative cells, three layers: inner cytoplasmic membrane, thin peptidoglycan layer, 5 to 10% peptidoglycan, outer membrane with lipopolysaccharide LPS, high lipid content, endotoxin LPS, lipid A, periplasmic space, porine channel, resistant to lysozyme and penicillin attack. Bacterial morphology. Bacteria have four shapes: cocky, bacilli, spiral forms, pleomorphic. Cocky, spherical, bacilli, rod, short bacilli are called scocobacilli. Spiral form, coma-shaped, S-shaped, and spiral-shaped. Pleomorphic, lacking a distinct shape like Jhalock. The different shaped creatures organize together into more complex parasites such as pairs, diplococci, clusters, strips, single bacteria with flagella. Bacterial morphology. So what are the names? Gram-positive. Start by remembering that there are 7 classic gram-positive bugs that cause disease in human, and basically every other organism in gram-negative. So we can see the shape, cocky shape, like this, round shape, bacilli or rod shape, spiral, comma, this is comma shape, this is S shape, it's a spiral. It's pleomorphic, This is clusters. And strips, this is diplococci. And with flagella, this one with flagella, this all diplococci. Okay, so of the gram-positive 3 cocci and other 4 are rod-shaped bacilli. The 3 positive cocci both have word coccus in their name. Streptococcus, enterococcus from strips of cocci, staphylococcus from cluster of cocci. So, 2 of the 4 gram-positive rods produce spheres that protect a dormant bacterium from the harsh environment. They are Bacillus clostridium. The last 2 gram-positive rods do not form spores. Corynebacterium, Listeracea. Gram-negative. Of the gram-negative organism, there are only 2 groups of gram-negative cocci. Both are actually diplococci, look like 2 coffee beans kissing. Here is look like 2 coffee beans kissing. So Neisseria and or Axella. There is also just a group of spiral-shaped organism, the spirochetes. This group include the bacterium Treponema pallidum, which causes syphilis. The rest are gram-negative rods or pleomorphic. So let's talk about exception. Mycobacteria are weakly gram-positive but stain better with a special stain called acid-fast stain. The special group includes organisms that cause tuberculosis, leprosy. Spirochetes have a gram-negative cell wall but too small to be seen with the light microscope and so must be visualized with a special dark field microscope. Spirochetes are all very slender, tightly coiled from the inside out. They have a cytoplasm surrounded by an inner cytoplasmic membrane. Like all gram-negative bacteria, they then have a thin peptidoglycan layer cell wall surrounded by the alveolus containing outer lipoprotein membrane. However, spirochetes are surrounded by an additional phospholipid-rich outer membrane with few exposed proteins. This is thought to protect the spirochetes from immune recognition. Stealth organism, axial flagella, come out of the ends of the spirochete cell wall, but rather than protrude out of the outer membrane like other bacteria shown in figure 2.1. Where is 2.1? 2.1, where are you? Figure 2.1. Figure 221. Oh, what was that? Axial flagella come out from the inside but rather than outer membrane like bacteria. 221. No, not here. Okay, figure 2.1 here. Cell structure: flagellum, basal body, outer membrane, peptidoglycan, inner membrane, and this is flagellum. So, okay, cell structure we will read here, but first we finish this chapter at first. So, okay, 2.1. The flagella runs sideways along the spirochete under the outer membrane sheath. These specialized flagella are called periplasmic flagella. Rotation of this periplasmic flagella spins the spirochete around the genium, where it thrust propelling them forward. Mycoplasma do not have a cell wall. They only have a simple cell membrane, so they are neither gram-positive nor gram-negative. Summary of morphological differences among the bacteria. 1 to 9. Morphology: gram-positive, gram-negative. So morphology about circular coccus. Circular coccus, gram-negative bacteria are Streptococcus, Enterococcus, Staphylococcus, and coccus or circular gram-negative bacteria are Neisseria, Moraxella. And rod bacillus, morphology rod bacillus. Gram-positive bacteria are Corynebacterium, Listeria, Bacillus, Clostridium, Mycobacterium, acid first. And gram-negative bacteria rod or bacillus shape. Enterics live in the GI tract, gastrointestinal tracts. Escherichia coli, Shigella, Salmonella, Yersinia, Klebsiella, Proteus, Enterobacter, Serratia, Vibrio, Campylobacter, Helicobacter, Pseudomonas, bacterioids, anaerobic, Haemophilus, border, Della, Legionella, Yersinia, Francisella, Brucella, Pasteurella, Gardnerella. Okay. Spiral morphology of spiral-shaped bacteria. There is no gram-positive bacteria, but it have gram-negative bacteria. Spirochetes, Treponema, Borrelia, Leptospira. Leptospira we can remember by Leptospira. Okay. Morphology of branching filaments, growth like fungi. It's have only gram-positive bacteria, no gram-negative bacteria. Actinomyces, anaerobic, Nocardia, partially acid first. And pleomorphic, no positive bacteria, has negative bacteria only. Chlamydia, rickettsii, no cell wall, without any cell wall. Gram-positive, gram-negative, both have no cell wall bacteria. Mycoplasma can be gram-positive or gram-negative, both. So morphological difference among the bacteria. This chapter is going to be finished, but so we have cytoplasmic structure, metabolic characteristic, oxygen, and carbon energy sources. Then it's going to be finished. Okay, sir. But let's take a break.